Disclaimer, disclaimer, disclaimer. Ladies and gentlemen, boys and girls of all ages, don't be bashful, don't be shy. There's no need to walk on by. No matter what you are doing at the moment, you will regret not joining us in the big tent and witnessing the wonders of the world in which you live. From the trivial sideshow tidbit to the paradigm-shifting high-wire act of freak show physics, there are things you will see and hear that have never been seen or heard before beyond the mystical laboratory research walls. You'll hold your breath in captive silence as Kirsten casually catapults herself high above the center stage, tantalizing us with tales upon the scientific trapeze. Thrill and delight as the dazzling damsel Blair braves daunting beasts of nature in her corner of the stage. Look on with astonishing envy as TWiT techs push buttons of flashing lights while juggling live feeds, unlevel audio, and multiple camera angles. And there will be a clown or two thrown in for yucks and such. So step right up, come on over, come on in. The big show is about to begin here on This Week in Science. Coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kirsten, Blair, and Scott.
Good science.
Tonight.
Yeah, good science to you too, Justin. Hello, everyone out there. This is This Week in Science. And yes, the visions that you have been seeing on the screen before you, if you are in fact watching the video feed of this, were members of the cast and crew and guests too. And I'm very excited about the fact that we have Scott Sigler joining us tonight.
Hello there. How are you?
Hello, we're fantastic. You look like you've got plants.
I do. I do. I have a bit of a green thumb. Grow the plants. Don't kill them very often, except in a story, maybe. But for the most part, they grow quite well. I brought something to share, though. I know that you guys are sciencey, but I have proof of the existence of the flying spaghetti monster because I washed my headphones and look at the resemblance. Look, coincidental? I don't think so. Proof.
Fine.
Proof.
Yeah, it's a sign of something. We have to get the flying spaghetti monster experts on that one.
I don't know.
The great
why we need
Randy,
an expert.
maybe.
I mean, the eye, it's all in the eye right there. I can see it myself. You can see it. It exists. So, yay. Go flying spaghetti monster.
Woohoo!
Trust us
FSM.
to come see it.
That's right.
Yeah.
FSM FTW. Well, anyone who has watched this show before knows that we've had Scott Sigler on a few times. And but if you haven't watched the show and you are not familiar with Scott Sigler, I just want to let you know who he is and what he does. He's an author, and he writes some really interesting material, science fiction, horror, and the science part of the science fiction is emphasized. So the books that he writes come from a very scientific perspective, taking science and that we know of currently, things people are discovering, and then using it, taking it that extra creative thought further to create a tantalizing reading experience, which can be a lot of fun. And Scott, what is going on in your book life right now?
The biggest thing going on in my book life right now is the launch of Nocturnal, which is my gigantic new hardcover, which is out April 3rd. People can learn more about it at scottsigler.com/nocturnal, but I've been working on this book since, I think, 2008 or so. It was originally out as a free podcast in first draft form and took a lot of feedback from the listeners and used that along with joining with the editors at Crown Publishing to make what I think is a really polished, very fun 80s buddy cop meets monster story which I kind of describe as Lethal Weapon meets Hellboy. So.
Nice.
All right, so we're going to talk a little bit more about the book in the second half of the show. We'll give you some more time to expound on what's going on there. But in the meantime, we have half a show to dig into some science news. And part of Scott's assignment was to bring some science news stories as well. You know that we all bring the stories we think are fun from the past week. So Scott's got a couple of cool stories on the docket. I've got a story about an unsolved scientific mystery that I'm hoping to get to today. And additionally, cancer's cured in mice. Again.
Great.
Again.
Again. Right. Exactly. Justin, what stories should we look forward to from you?
Oh my goodness, what did I bring? That's a good question. I have oh wait, I'm on the wrong thing. I think I've got fetus in my brain. Fetus for brains. Is that possible? Could that be a real story?
I don't know.
Could be. Planetary paternity test took place recently, and a spontaneous coherence or seeing patterns in an altered state of matter.
Interesting. And Blair, what's going on in the animal corner?
I have a story about birds and how we all know in the northern hemisphere they fly south for the winter, but where do they fly when climate change hits? Some interesting info from the Christmas bird count to talk about.
Fantastic. Fantastic. All right, so let's dive into the science news for today. All right, one drug to cure them all, if you are a Tolkien fan, right? One of the holy grails of cancer research is the search for one drug that could possibly cure all of cancer. I mean, you hear it all the time, looking for the cure to cancer. Well, the more and more we hear about cancer, the more it seems to be a very complex beast that's not necessarily something that could be cured by one drug, that there are microenvironments and lots of things that go on in different tissues that don't happen in other tissues that make the treatment of particular types of cancer need to be very specialized. So the idea of a single cure to cancer has often been kind of, as we look at more and more at cancer, thought of as a harder and harder thing to reach. But maybe not. There is a biologist, Dr. Irving Weissman of Stanford University School of Medicine in Palo Alto, and he and his team about 10 years ago discovered that leukemia cells have a protein on their surface called CD47. And CD47 is like a signal to the immune system to leave the cells alone. And so normally, if a cell is a cancer cell, that means it's growing, splitting out of control. And the immune system, something's broken on the inside of those cells that are growing out of control. And the immune system is tuned to find those breaks and to respond accordingly. And in the case of this particular situation, these leukemia cells 10 years ago, CD47 basically was a white flag saying, leave me alone, immune system, leave me alone. And so that's why a lot of tumors are able to grow unimpeded by the body's own natural defenses. Now, they've continued to look for something that could block CD47, and they've come across, they've got an antibody, they've got a blocker for CD47 now. They're able to block it, and they found in laboratory tests of multiple tissue types, lots of different tumor types in different tissues express CD47 more so than normal cells. And so if you block the antibody, if you block the CD47, that allows the immune system to get in there and attack the cells, kill them, and stop the tumor from growing. And they showed in the laboratory that if they used this antibody, that they could stop the growth of tumors. They could cure tumors in petri dishes and in mice. So the next step is to find out whether or not it'll work in people.
Which would be a very good thing. I'm like so jaded at this point. because we've seen so many of the stories that show the studies that show promise that for whatever reason can't get proven in the either a great ape trial or human trial later on. But you know what? If it's gonna happen, it wouldn't have happened in the past. It would be happening now, right? Because we haven't found it yet. So it's not that the answer isn't out there. It's not that the cure isn't out there. It's not that we can't find it. It's just that we've been burned a few times. But let's keep all of our collective anti-cancer fingers crossed for this one, because this would be really cool.
It would be very cool. But the interesting thing about this, it's taken them 10 years from their initial discovery of the CD47 protein marker, this receptor. 10 years, an entire decade for them to get to this point of being able to come forward and being confident enough in their research that they could write a paper, publish it, get it peer reviewed, publish it, and be confident enough that it could lead in the direction of some kind of treatment. I mean, it's not like it was like a year ago or a couple of weeks ago that they started working on this. They've been working on this research for years to be able to get where they are.
Yeah, that's a really good point. I mean, these things do take time. And it's one of those things, too, that we do notice in just about every story that we've covered related to cancer, is there's always a sort of qualifying disclaimer in there somewhere where they're saying this could possibly lead to, may lead to, potentially has the maybe ability in some point later on of unlocking the secrets that we need to unlock in order to find a cure for cancer or to cure certain types of cancer or and, you know, other diseases are in that category, too, of finding those sort of baby steps. And, you know, they follow them. You know, they don't just follow like, oh, this could be something interesting. Well, that's nice. Let's find something else. No, there's people out there pursuing all of these lines of potential cure. If it's gotten to the point where, I mean, those tests sound like, you know, they have some real world examples of it working. But let's just hope it translates into humans. And the reason we use mice is because a lot of the stuff is going to translate pretty well.
Yeah, they've already received a $20 million grant from the California Institute for Regenerative Medicine to be able to take it to human studies. And Dr. Weissman, according to this article at Science Now, says we have enough data already that I can say I'm confident that this will move to phase 1 human trials. So He's got a lot of data. He really thinks he's onto something. And so this wouldn't necessarily be, you know, the only thing that you'd have to take to cure cancer. It would probably be a drug that would be used in combination with chemotherapy to be able to remove, to be able to beat the cancer back into remission, or to stop cancer from spreading from the primary tumor, metastasizing to other organs, other tissues. But it's definitely, if it works out, fingers crossed on this. Fingers crossed this could become a very powerful tool in our arsenal for helping a lot of people, which I think is really exciting. But you're right, Justin. We've been burned so many times before.
We have, and I'm very hopeful because, I've lost a lot of people close to me due to cancer, and I expect to be getting it at some point because of the way
because you won't
I have
stop
lived
smoking.
my life. Oh, right. So I'm really hopeful. It's not that it's like, cancer, who cares? I'm not trying to be flip about it. I am extremely hopeful that this works. I just, I just hope it happens. If it doesn't happen in my lifetime, it could still happen in 20 years. Right? So I think we need to find a cure because I didn't live my life responsibly.
Happening in your lifetime is kind of the key phrase there because of it.
Right. Which could be,
first, then not in your lifetime.
I know. I don't know if that's 10 years. I don't know if that's 20 years. I don't know if that's 30 years. But it really would be nice for it to happen in my lifetime. That's going to be important. I'm pretty sure.
Yeah.
It isn't now. I'm fine now. I'm cancer free for all I know. I'm doing great.
For all intensive purposes.
For all I know, everything's fine now. Don't anybody worry. But yeah, it's gonna happen. It's gonna end badly. That's just it's there.
High pessimism.
Well, I mean, I smoked. I worked like a decade at a car dealership, which is like basically like working in a parking lot, sucking fumes all day. And there's enough genetic history out there. It's coming.
I'd forgotten about the eternal optimism of this show.
I know,
It's
right?
been a little while, and I just needed a pick me up today, and always count on Justin for that.
Well, speaking of optimism, I was sitting back here thinking, and I mean, it'd be great if we cured cancer. I'm all for it. But of course, in the back of my head, I'm thinking, if we cure cancer, won't that boost the population problem just beyond the charts? Wouldn't that be?
No, because then we have war. We've got war for that. Don't worry.
Oh,
We'll
okay,
just
all
blow
right,
some
all right.
people up.
Scott,
We're good
she's
then.
There's always
too
war.
young
Okay.
to sympathize with old people. That's all it is. She doesn't understand yet.
Hey, no, I understand. It's the scientist in me is thinking about carrying capacities and things like that, and I just
The
can't.
biologist. Yes. Population dynamics.
Yeah.
All right, Justin, what did you bring for science? Let's move into. Did you bring something more positive, uplifting? Maybe we can,
Actually,
whistle
this
a
is
happy
pretty
tune
wild.
for
This is
Scott.
using fetuses as brains.
Oh, great, awesome! It's like the opposite direction.
Yeah, this is the headline here. Your inner child may be closer than you think. It turns out that the same genes and proteins that promote growth and development in embryos stick around and show up again with a new function later on in life. They play a role in sending the chemical signals that help adults learn. That they remember, they're part of the system that helps, the signals that help us remember things or even forget things, depending on their strength and weakness, and maybe involved in addiction, as well as whether they're strong or weak. If they're a little bit weakly active, could be present in even some mental disorders. So this is a quote from Professor Andres Villu-Maric. I'm guessing it's M-A-R-I-C-Q. It must be a foreign language. We found that these molecules and signaling pathways do not retire after development of the organism, but have a new and surprising role in the adult. They are called back to action to change the properties of the nervous system in response to experience, which is, I think, a really pretty amazing thing. So he has another kind of quote. The adult nervous system is not a stagnant tissue, but rather dynamic and plastic, with the strength of synapses, specialized neuron-to-neuron connections, changing with experience, learning, and memory. So it's not a fixed thing, like when you're done making a heart, you're done. There's the heart. It's a continual system of growth I think it's kind of interesting, too, that it's the same pathway that initially sort of lays out where all of our organs are going to be. So it's not a dedicated, brain growth pathway, but it's something that's throughout our whole nervous system and sort of dictates, okay, the lungs are going here. We're going to put a liver over there, a couple of kidneys back over here. And then it was thought that was all the only function it can have, but now they're finding these are the WNT genes. And proteins that they produced are also called WNT proteins. Then they do this thing called WNT signaling, and they're heavily now involved in human learning, memory, and even forgetfulness. And so this study was completely done on worms because their nervous system is a good model for our neurons, I guess. It's a similar, very basic system that they can study. And they put in some of those glowing jellyfish genes so that they can
I love the glowing. Yeah.
Isn't that awesome?
Glow, jellyfish, glow.
Yeah.
I see your brain.
He's quick to emphasize here also the WNT chemical signal is different than the actual nerve signal carried by a neurotransmitter, which in the new study was acetylcholine.
Acetylcholine. Acetylcholine.
That's the one. The WNT signal is a secondary signal that, so you have the main neurotransmitter chemicals are making the connections, but the WNT signal controls sort of the volume of the neurotransmitter signal. So it determines how sort of loudly it's connected or how quietly it's connected. The more quiet the connection, the less of a connection, the less able you are to access it, so there's less of a memory. The louder it is, the more readily accessed it is. And so you can think of something like an addiction. If they're being triggered to be very loud, then you're thinking about that thing you're addicted about in your head very loudly. So they did some stuff. They crippled some various genes in worms, and they identified the signaling pathway by which the WNT protein in one nerve cell sends a chemical signal to another, telling it to increase the number of receptors on its surface, and thereby increasing the strength or volume of the nerve signals between the cells. So pretty cool.
Yeah, I think it's really. I mean,
and
oftentimes
this is also,
we think of genes that only work for development. So you have certain genes that get the brain to turn into the adult brain, develop from the fetal brain into what it will be as an adult. But then once you're an adult, there's been this idea that completely different genes rule the functioning of the brain. So it's interesting to think that the plasticity of the brain, the ability of the adult brain to not remain static, to enable it to learn, actually relies on these developmental genes. So
that and the dual purpose of these genes too. In the initial, they're laying out sort of a set structure of where the organs are going to be, and they're very important to our early stage growth and development. And then it's sort of like they have that's a very important responsibility. And then they also carry that responsibility into our mental evolution based on behavior and experience, right? I love the fact that those two things are connected, and somehow our bodies have those prioritized the same. You know, you have to have lungs, you have to have a liver, your liver has to go here, it has to be connected like this, and you have to have the proper reaction to something scary or something good, right? Like you have to, those are equally important. It's also that this is one of those two things that's like, it's the beginning stage. It could possibly potentially lead to new forms of treatment for schizophrenia and a number of other mental disorders. Because if you can really figure out the pathways by which these signals are going through in the human brain, you can control volume a little bit better. And in some, certainly in schizophrenia and understandably in a lot of mental disorders, it's the volume of the data that's vying to get pushed to the forefront that is causing a lot of the distress and a lot of the problem.
Yeah. And this is all from experiments in worms.
In worms.
I know. We can potentially make inferences about ourselves from the experiments with worms. I mean, scientifically, it all comes back to, these were just experiments in worms. The same way that it's like, cancer was cured in mice. It's not an experiment in people, but at the same time, this is a signaling pathway that probably has been has been preserved across species, so there might not be a lot of a difference in the human brain and the adult brain. So they believe that it's going to be involved in human learning based on
what is likely the next step for them with this? They go from worms to what? To start testing to see if it's consistent across different species.
Yeah, so from worms probably to mice. Because mice are the most closely related aside from primate species of the lab organisms that we use. So it would be worms, mice, and then possibly looking for stuff in primate brain tissue, human brain tissue, and then possibly looking for evidence. I mean, if you can, they're not to a point yet to be able to do this kind of genetic research or protein research in humans. So that's a long way off.
But perhaps they may be able to develop drugs based on the idea that this is going to work in humans, and it won't have any lethal side effects or anything. It won't be something that you're doing, like a lot of the cancer treatments do a lot of damage to the body. So it's hard to get a human trial because you're doing damage while you're attacking the cancer. But if this is something that can be tried without having other damage to the system as a alternative medication for something like a schizophrenia, then it could get pushed ahead, I think, that way without too many trials. I guess it just depends on on how much potential damage something can do going forward. Because if it's another sort of antidepressant or antipsychotic, I can see them doing this without having to do an antipsychotic trial in mice.
Right, but and the real take-home message of this right now is just giving us information about the pathways that the neurons use, that the brain might use to be able to allow learning, addiction, what might to take place, and what might get messed up in certain psychological or mental issues. So, it's kind of this is kind of a this is how it works kind of study, letting us know how it works, but it's interesting. Scott, I think you're up.
I have a story for you. Now, not being the hard-hitting science type like the two hosts of this show, I've tried to find things that are more suitable for what I study. So, do you want to be creative? Science says, booze it up. This is reported in Science News, volume 181, from March 24th. Getting a buzz from booze may boost creativity. Men who drank themselves tipsy solved more problems demanding verbal resourcefulness in less time than sober guys did, a new study finds. Sudden intuitive insights into tricky word association problems occurred more frequently when men were intoxicated but not legally drunk, says psychology graduate student Andrew Jarosz of the University of Illinois at Chicago and his colleagues. And he was probably drunk when he said that.
Probably.
Took a more deliberate approach to this task. A moderate alcoholic high loosens a person's focus of attention, making it easier to find connections among remotely related ideas, the scientists propose in their publication in Consciousness and Cognition. In the study, 20 social drinkers watched an animated movie while eating a snack. Volunteers then drank enough of a vodka cranberry drink, could be a wine cooler, which could color the study one way or another, to reach an average peak blood alcohol level of 0.75, just below the current 0.8% cutoff for legal intoxication in the United States. Another 20 social drinkers watched the same movie without eating or drinking. Men in both groups then completed a creative problem-solving task, and they had to find a word association. They were given 3 words and had to find one word that matched all 3, say peach and arm and tar, and then had to figure out that the missing word was pit. On average, at peak intoxication, which I think is a good phrase for any study, when you can in your study say at peak intoxication, it took them an average of 11.5 seconds to solve the problem, compared to those who were sober, an average of 15.2 seconds, and both groups compared the same on tests before the study began.
Wow.
So I think it's kind of awesome that there's
awesome.
A little boozy science, and finally something that says, hey, maybe it's not all bad. Maybe it helps you figure out connections to things. And the study goes on to say that there may be more deeply reaching implications for people who work on the same kind of creative process over and over again to do something that could help them make better associations or different associations. So having a couple definitely seems to increase creative connections, I believe, but then you have to sober up in order to manifest those connections and to finish off whatever the spark that creativity gives you. But I think that's solid science right there.
If you ever watch Mad Men, though, yeah, they all have the little mini bar in the office. And they have other people who implement the creative idea, have to actually do the artwork or, come up with the print. So that's really all they're there for, is to come up with the spark. And I'm glad to know it doesn't hinder your ability to make associations between things and be creative.
But the key here is that they're not completely intoxicated. So this is just below the official intoxication level, not actually officially intoxicated.
It doesn't say if they went further to study, like, okay, let's say 0.005% below the intoxication limit, and then next phase is 3 sheets to the wind. It didn't actually say if they tested what happens when you are absolutely blotto. Who knows? That'd be an interesting graph to see. Here's your creativity level slightly under the intoxicated limit. Here's your creativity level when you are really blotto. And here's your creativity limit when you are throwing up in the toilet, because I think those would be different.
And I
There's
wonder
going
if
to
they
be a huge
limited it.
difference. Yeah.
I wonder if they limited it at the just below the legal drinking limit there on purpose, so that people wouldn't have an excuse like, look, I had to do a creative writing exam.
Yeah.
I had to put down the extra couple of beers because I'm even better at 1.0. So,
and going back to the Mad Men reference, there is a huge opportunity here for some beverage company to further fund this study and then be able to say, Bacardi makes you smarter. It's proven by science.
totally.
I would be interested to see if they got the same results with a written exam, just because I'm wondering if they're coming up with the answer faster or if they're deciding to say it faster. Because when you're sober, maybe you're triple thinking yourself before you speak, as opposed to just coming up with an answer and saying it because you're more...
I like where your head is at. I think that's a distinct possibility, because having experimented along these lines on my own with some unverified studies, yes, you do tend to say the first thing that pops into your mind and say it a lot faster. So it could just be a limiting of inhibition allows you to not second guess yourself when you're coming up with the correct answer right away.
Yeah, I think that's a very interesting point. All right, we're coming up to the end of the first half. Blair's Animal Corner.
Well, it's not a super fun topic today, though. It's a little sad, actually. So climate change and animals, they don't get along that great because, when your climate is changing, that means everything in your habitat has to change. And so it's kind of a common thing with animals of studying them moving in relation to climate change and seeing if an animal can move at the same rate that the temperature and the plants are changing around them and all that kind of stuff. And so they do every year, they do the Christmas bird count, which I've participated in a couple of times. It's very fun. You walk around, you do your birding, you take numbers of all the different species that you see and in what places, and you send it off, and they compile the results. And so over the past 35 years, they have actually seen animals move, the birds move, and catch up to the temperature change. But it's taken them 35 years to catch up with their temperature change. So they found, looking at 59 bird species, most of them were moving in relation to the weather. And their example that these were black vultures, and they spread north in the last 35 years, which makes sense because if you think about the tropics, basically as climate change happens, the tropics are expanding. And so they need to run away from that to try to catch up with their temperature that's moving northward from them. And so they're moving northward, and they have gone all the way up to Massachusetts, and they used to be in Maryland in 1975 at the same time of year. And when they were looking at temperature changes, the temperature change is it correlates exactly with what it was in Maryland in 1975. But of course, their other example was that the red-cockaded woodpecker, which is endangered, hasn't moved. But that's because they have a very specific food source that hasn't moved yet. So even though birds can fly, so they could move pretty easily, they can't move as easily because their food source isn't moving, and a lot of their other resources aren't moving at the same rate that they could. So it's taking.
Gotcha. So they're showing up in Massachusetts now, but the butterflies are like somewhere in between when they're showing up at the right time, right? Or whatever their food source is the timing isn't quite there yet.
Right. So some food sources could move faster, and some couldn't move as fast as the bird would like to move. The vulture's kind of an easy example for an animal that could move because the vulture's going to eat anything, so they can go anywhere that's dead things.
Dead.
Yeah. But so basically the implication with this study is, okay, yes, we know that the climate is changing, if you want to believe that. Yes, we know that animals are responding to it, or at least attempting to respond to it, but it's taking them a long time to respond.
They're not responding as quickly as we would hope they would be able to.
Right. And
it's not instantaneous.
It's a domino effect. So they can't all move at once either. It's not like they're all packing up their bags and going, let's go north, guys. That takes time.
Yeah.
So what's going to be interesting to see is if temperature continues to increase at the same rate it is now, there's a chance the animals could continue to catch up. But at the same time, if it goes faster, it's not looking great for these animals. It took them 35 years to adjust from the 70s, when this stuff was just getting started pretty much before, the spike had begun fairly recently in the global warming and them seeing animals move for global warming. And so if it increases more exponentially, it's going to be really difficult for the animals to stay chasing that temperature gradient.
Right, because like you mentioned, it's not just the temperature gradient; it's their food sources. Right. It's the right temperature and the right weather environment for them to raise their young or to winter, whatever. Right. There are so many factors that play into it that if just moving. The trees start moving north, the weather the temperature changes, the trees move, the birds have to move, the insects move, everything has to follow, and it doesn't happen fast enough.
Mm-hmm. And when you think about it, birds, they can fly over things like bodies of water. Mm-hmm. But for example, this black vulture,
that's a big jump for a frog.
Yeah. If suddenly you hit, water and you can't go any further north as an animal, you're kind of in trouble.
Mm-hmm. Can't go under it.
Which could be either a silver lining for the birds or completely disastrous, depending on, because I'm thinking of the potential predators, the egg thieves, the things that might normally prey on the birds. As if they can't follow them, they could end up in a less predatory area, or they could move into an area rife with predators. It's kind of a crapshoot there.
Well, but then you come back to my friend carrying capacity that we were speaking about earlier. And if you don't have any predators, you're all going to die anyway.
And she didn't say Karen capacity. She said carrying capacity.
Sorry.
Carrying capacity.
Carrying
capacity is a whole nother character.
K.
Carrying capacity. Exactly. And as Socrates 2020 says in the chat room, what's interesting is that the people haven't got a clue what to do. But you know what? Socrates, as Bill or Ted would say.
Excellent.
Excellent. People are thinking about what to do and people are trying to work on it. And, maybe we all just need to go nocturnal. And that's what we're going to talk about when we come back after the break. It's time for the break. Scott Sigler is our special guest, and we're going to hear more about his book, Nocturnal, that's coming out here very soon, and have some more science stories for you after the break.
And we're back with more This Week in Science.
That's right. And before the break, I told everybody that we were going to dig into a little more about Scott Sigler's new novel, Nocturnal. Tell us a bit about what's the plot? What's going on with your new book?
Well, the plot has given the publisher fits as far as how to market it, because it starts out like a police procedural. You got it's kind of my tribute to 80s buddy cop movies. So you have Brian Clauser and Pookie Chang, two San Francisco homicide inspectors who are on the trail of a series of particularly brutal serial killings. And then Brian starts to have dreams that are very similar in scope to the serial killing. And then when they arrive to find a new body, they find that his dream has enacted that almost exactly. So he quickly falls under suspicion as the killer, and it's up to him and his partner to kind of figure out what's going on. And then it takes an utter left turn, gets into the hard science and hypothetical science area, and largely turns into more of a Sherlock Holmesy type cult mystery mixed in with some really high violence, monstery type goodness. So again, the best way to describe it is Lethal Weapon meets Hellboy. It's really out there, and there's a lot of. I'll talk about some of the cool science that is in the book if you like.
I would love to hear. Yeah, tell us a little bit. You mentioned that it takes a hard turn, and there's science in there. What is it?
The science is. It's kind of looking at some divergent evolution of humanity and trying to hypothesize if something had gone really different, if the strain of humans had started to become something completely unique, but then the what was the phrase she used? The population level or the carrying capacity. The carrying capacity, say, is already full, and when people realize that in their own primitive way that speciation has occurred and that this thing is a threat, they gang up and want to destroy this new strain of humanity. And that pressure, that societal and evolutionary pressure, then forces this new strain into some heavy changes in order for them to survive. And those changes are a little bit in the stealthy category and things that will allow this offshoot of humanity to survive and live amongst us, but still keep their own culture and their own form of breeding. It gets a little bit different because what I'm trying to explore is if the eusocial structure was applied to humans, if you had a breeding queen and a breeding king, and how could that apply so that they do look like us and act like us, but they are extremely different, and how do they stay hidden and how do they stay safe? And that leads back into the mass murder concept. And I think the funnest part of this book for the sciencey-minded people who listen to this show, watch this show, everybody knows about the Punnett Square. And
right.
Nocturnal, this is Nocturnal. It is extremely girthy. There's a lot of girth to this book. It's big. in this strapping, girthy book, there's my favorite scene is a Punnett Square analysis where there is an additional sex chromosome. So instead of it being 4 squares, it's 9 squares, and every square, every combination of these 3 sex chromosomes produces a different permutation of what these things can be. And it's very fun to go through and start to imagine the combinations that I actually show you in the scene that you've seen before through various genetic analysis of bodies and residue left on bodies. You can start to put the pieces together. This combination equals this combination equals this, and then there are combinations that you have not yet seen, and that leads us all the way towards the great big finale of the book. And I try and
wait, don't tell us how it ends. No, it's spoiler.
I won't.
Don't tell us the ending of the book, Scott. That was close.
I love spoilers. Come on.
I just try and write big summer blockbuster type novels that you would see in a theater. So the ending is completely over the top. It's a ton of fun. It's very bloody and violent. And if you guys like movies like Lethal Weapon and or Hellboy, Predator, that kind of thing, you'll eat this up.
Sounds like a lot of fun. And there is actually, this is a book that we're talking about, but you have a trailer that we can watch, listen to. So let's do that.
I hunt. My brothers and I find the weak. Those who won't be missed. We stalk. We catch. We feed. After each kill, we draw the symbol to stay safe. The darkness is hours. I always wake, horrified at what I've done. Wondering why I still feel the blood on my tongue. Sickened that it tastes so good. Oh God, this can't be real. I no longer know whether I'm a man dreaming I'm a monster, or a monster dreaming I'm a man.
Awesomeness.
Okay, so what are the dates on this? You've got the book coming out next week through the publisher.
Yep, the book is out April 3rd, and it should be in bookstores, although it's impossible to tell these days with the current state of publishing if it'll be out or not. It is available for pre-order at Amazon.com, Barnes & Noble. Links are there at scottsigler.com/nocturnal. And if people are interested in an autographed copy, they can order from Borderlands Books in San Francisco, which is borderlands-books.com. They have an 800 number there. I go down to that store all the time, sign copies, and they send them out. So people should check it out, and it's also available as an ebook. But if you can buy the hardcover, because I'm trying to crack the New York Times bestseller list again.
Good luck with that. And if anybody is interested, are the Nocturnal podcasts still available? Because this is something that you worked on. Like your whole process usually is to work on books and get feedback from your audience and then fine tune the books. Is this something that people can listen to?
No, we pulled down the podcast. It was up. It was free for 3 years. We pulled it down once we started hardcore work on the final draft. So I podcasted as I wrote it, write a chapter, then record it, podcast it. Next week, repeat the process. And so what I podcast was really fun and people loved it, but it was a first draft. And there was still a lot of work that needed to be done. So now the story has changed so much that we pulled down that audiobook so that it wouldn't cause a lot of confusion. And eventually, when we re-record this and get it out as an audiobook, we'll get the free podcast back up. But for now, ebook and hardcover is the only way to get it.
Very cool. Very, very cool. I'm looking forward to getting my hands on a copy and taking a listen. I think it'll be a lot of fun. But let's do some more science news before the end of the show. We have more stories, right? There's more science. Mm-hmm. How about some unsolved mysteries, right? We can talk a little bit about the mystery of pine mouth. What? Exactly.
Pine mouth?
Yeah, what? Probably never heard of this, actually.
Never.
Yeah, well, it turns out that there is a syndrome called pine mouth that occurs within about half a day to 2 days after eating pine nuts. And according to the FDA, the U.S. Food and Drug Administration, this has been complained about. It's a bitter metallic taste that's associated with the eating of pine nuts. And they call it pine mouth. It lasts between a few days and a couple of weeks. So if you've ever eaten pine nuts and had this bitter metallic taste that has just kind of entered your mouth and stayed there for a couple of weeks, this might explain it. The question is, what aspect of pine nuts is causing pine mouth? And so researchers have been trying to find out the causes of pine mouth for several years now. A brand new study has been done by some researchers publishing in the American Chemical Society's Journal of Agricultural and Food Chemistry, and they can't answer the question. So So it's still an unsolved mystery. This is still something that nobody really understands, except they do now know that it is the result of different species. So there's one particular species of pine nuts that seems to cause this bitter metallic taste, whereas other species do not. The only problem is, so the species is called Pinus armandii, and so this one particular species is to blame. The problem is when you buy pine nuts, you're not buying a single variety, a single species of pine nuts. When you buy them, you're buying a mix because the nut dealers get nuts from all over the place. They mix them up. They just sell a batch of pine nuts to the grocery store, and you're getting pine nuts from all over the place. And so you could have those Pinus armandii nuts in a batch at any point in time or not. And it's very hard to tell. So the researchers are trying to they're looking at the fatty acid diagnostic index of the pine nuts and DNA analysis in order to be able to figure out what pine nuts are in which samples. And so this might be able to find which nuts are most involved, including the Pinus armandii. But they don't know. The fatty acids they thought might be related, not related. They have no idea. Pine mouth, as yet unexplained, just like the Loch Ness Monster.
Could the two be related?
Very possibly.
I think
Especially,
Loch Ness Monster could be responsible for the pine nut issue. Have we considered this?
I hadn't considered it, but now that you've brought it up, I mean, it's possible. Especially if you're eating pine nuts in Scotland.
I feel that until you can prove that it's not true, then we have to accept it as a valid possibility.
That's right. It's a valid possibility. I think you're very right. But yeah, pine mouth, have you guys ever experienced this?
Nope.
No.
I really haven't had many occasions to have nuts in my mouth, and when I have, I don't recall metallic.
Pinus in your mouth, dude. Come on.
Pinus nuts, Pinus and nuts, or what? However, it's I wouldn't know.
That's not where I was going with. Okay, moving along.
I don't think I'm a good. I'm the guy who eats all the leftovers, and if the milk's 3 days bad, it's still, I paid money for it. We can still have that. So I don't have many reactions to different food stuff. So I've never experienced anything quite like it. But pine nuts are good.
Pine nuts mostly good. I'd love to find out more about this syndrome. All right, Justin, tell me another story.
That before. Okay, so for a long time there's been debate. Or actually, it's not even been a debate. It's sort of been assumed that our moon came from a big chunk of the Earth that broke off when we got impacted by something really big sometime around 4.5 billion years ago. But there was a recent chemical analysis of moon rocks, lunar material collected back from the 1970s from the Apollo astronauts. And it seems to be contradicting that hypothetical, saying that what we found is that the child does not look any different compared to the Earth. The child in this, of course, being the moon. It's a child with only one parent, as far as we can tell, says Nicholas Daufus, associate professor in geophysical sciences at the University of Chicago, co-authored the study. So what they were looking at titanium isotopes, and basically figured out based on this that our planet, it should have a mix. If there was some sort of, and the original theory is it's a Mars-sized impact. It's something really massive that hits the Earth, breaks off a chunk of it, and then goes to the moon. The only thing that kind of confuses me about this whole thing is that if something as big as Mars impacted with what was forming as Earth back then wouldn't those two have merged significantly enough that you wouldn't see like a whole lot of difference between the new Earth, big giant asteroid thing, and the moon?
Well, I mean,
So-
it could have merged with part of it, and part of it could have been ejected from the collision. So the impacting body doesn't necessarily have to remain in a hole. Part of that then eject and wind up forming into the moon?
Right, and the idea is it would have to be, the part of the object that hit us would have to be on the moon, and they say that doesn't look like that because everything there looks like it came from the Earth. So let me see. This is when we look at different bodies, different asteroids, there are different isotopic signatures. It's like they're different DNAs. It's saying that meteorites, which are pieces of asteroids that have fallen to the Earth, contain large variations in titanium isotopes. Measurement of terrestrial and lunar samples show that the moon has a strictly identical isotopic composition to the Earth. So I guess that makes sense then, because the Earth we'd already have that amalgamation accounted for. We wouldn't have an amalgamation accounted for. It's just completely identical to us. So while we thought the moon had two parents, it looks like it's just a part of the Earth. Now, the question then is it just a part of what was forming to be the Earth? I guess that's the next step. What was forming what eventually became the Earth, some of it ended up stuck out there in an orbital area and coalesced to become the moon. So that means we co-evolved. I guess we wouldn't even be a parent at that point, right? At that point, we'd be siblings?
Virgin birth.
Yeah, like a virgin birth. Parthenogenesis.
Immaculate moonception.
We're talking about lunar parthenogenesis, or even budding, like yeast.
Ooh.
That's right.
Awesome.
The moon budded from the Earth like a baby yeast. Yeah, well, it's interesting to think of, if something hit the Earth, would that object have mixed enough with the Earth so that that object would have ended up part of the moon as well? Or would it have been just like, when you think of a collision, the typical billiard ball example, a billiard ball hits a billiard ball and the other one just shoots off. And so would it have been just a piece of the Earth that shot out and then was continued to be captured in our gravitational field, and that became the moon?
Right, and I think that was the idea, is that the impact created like a giant debris cloud, like a massive debris cloud into our orbit that then coalesced into the moon, as opposed to, getting like a billiard ball getting knocked out of the way or anything like that. Mm-hmm. Which I guess means both bodies, though, if it's the size of Mars, Earth's a pretty good size, pretty dense, but Mars is pretty good size. I wouldn't want to take Mars big. On. And
Mars is no punk. Mars is no punk. Mars has got some size to it, and it will mess you up. So.
It's got some size, but it's got density issues. One thing that we've proven over and over again is that the Earth is by far the densest planet in our solar system. Which I don't know.
We got that belly full of molten iron. Is what we got.
Yeah.
Belly full of molten iron.
So does Mercury, but Mercury's smaller.
How do you like it? Yeah.
How do you like it?
How do you like that, Mars? I don't think you like that very much at all, do you? But we got iron and you don't. So
Mars is molten. You got rock, nothing.
You're all rock and ice. You got nothing on Earth.
This is interesting. I'm thinking of this study also in terms of what was it? The big. There was another idea that there were two lunar bodies, and so that kind of goes along with the idea of there being a cloud around the Earth, and that cloud might have coalesced into a couple of bodies, and then maybe those bodies might have run into each other and become the moon over time, right? And they could potentially all be made of material from the Earth. But then if we'd run into this giant Mars-sized object, shouldn't we find evidence of that Mars-sized object, Theia, in the isotopic signature of the Earth? Do we find that already?
Well, right. That's the thing. I guess it's, if it's buried, if we swallowed it up, right, and it's like maybe we melted it down and turned it into our iron core, right?
Thea is our
Then
core.
Yeah,
then
that's
we don't
right.
know.
Yeah, you come into our orbit, boy, you're going to get melted down and put into our core. That's right. That's how we roll on Earth.
But we should still, but the idea there is we should still see some element of it out there in the moon, I guess. There is a little, a slight, still you can keep the debate going on infinitum, because it's possible that even though the titanium is refractory in the way it may have gasified in the giant impact and then become incorporated into the disk of the Earth's orbiting material that developed into the moon and might have erased signatures of the titanium from Thea, which is Thea, Theia, is the what they call the Mars-like object that may or may not have actually bumped into the Earth.
I'm going to go with the gasification idea. I think that's what happened.
I might be slow on the uptake here, but what the study is saying is there is no Thea. Either Thea smashed into the Earth and all of the material of both melded together to the point where they're undifferentiatable, or there was no Thea and something else happened. Is that what the study's saying?
Thinking, yeah. Basically, it's the hypothesis right now is that this is one piece of evidence that could eliminate the Thea theory or Thea hypothesis. Because if all these titanium isotopes are identical to the ones on Earth and don't really look like they came from somewhere else in the solar system, chances are they didn't, and the moon isn't a chunk that got knocked off of the Earth, but may have in fact formed the same time that we did of the same material. Or it means there's another idea that they have to figure out. Maybe the Earth formed and was doing really well, and then 4.5 billion years ago blew up just spontaneously and threw out a big chunk of moon. And maybe it could happen again.
Yep.
Spontaneous moon burp. Great.
Spontaneous planet explosion.
That's right. All right, Scott. Drive us home with the last story of the night.
I will. Keeping in the vein of our pine nut story, and since we're talking about eating and food and digestion and all of those things, and moon burps, are you blind and are you hungry? Then science says you should drive to Taco Bell. This was reported in New Scientist today, actually. Google's self-driving cars this week passed a new milestone after the web giant asked a blind man to take the wheel, which is kind of mean when you think about it. They posted a video that shows Steve Mahan taking a trip to a Taco Bell restaurant after Steve got really, really baked at about 2 in the morning.
Hey, no, I thought he was just blind.
He was, well, it's, to deal with the glaucoma, right? He's 95% blind, far beyond the legal limit. Mahan, who is the head of the Santa Clara Valley Blind Center in California, made the journey without having to touch the wheel or pedals, thanks to Google's driverless car technology, which uses an array of video, radar, and laser sensors to constantly read the road. The car in the video followed a pre-programmed route, successfully navigating junctions and corners without incident, and Google's autonomous driving fleet has racked up more than 200,000 miles of driving, including unplanned journeys on public roads.
Doesn't that make you feel great?
Trips to Taco Bell, and many, many trips to Burger King's all-night window.
Awesome.
Yep.
Yeah, how many of those 200,000 miles were racked up with fast food drives?
Come on.
Late at night.
Thing ever. It's 3 o'clock in the morning. You're a little hungry, but you're beyond the legal drinking limit because you've been doing that vodka study with creativity. And now you just got to get some food in your belly. Thanks to Google, you don't even have to be at risk. You can just go to Taco Bell and you can chow down. It's really this is a big development, I think, for everybody.
I think it's a really interesting development. I mean, the whole idea of the driverless car or the autonomous car, the car that you don't have to do anything, just sit in it and it takes you where you want to go. It's fascinating. It's like having a chauffeur, right?
Well,
but it's a robot.
It's not just that, but if we've got this going, then we can start to get the other technology where the cars are kind of talking to each other and lining up more like cars in a train than what the traffic we see in the highways now, which has drastically reduced drag, increased fuel efficiency, and has the potential to get rid of gridlock almost altogether, because a lot of cars line up and operate as a single unit, accelerating at the same time, braking at the same time. And our cars kind of become their own form of mass transit. And there's a lot of good things that can happen in it.
That may all be well and fine, but yeah, 24-hour tipsy taxi. That's what I like. There's brilliant.
No question. And there's the concept of freedom of movement for handicapped people and visually impaired people is really exciting from the serious perspective. But from the silly perspective, I mean, Taco Bell is not making a commercial out of this is just crazy.
Well, there's people who are disadvantaged for whatever reason. They can't drive, and the elderly who've had their licenses taken away, and the young kid who, like, the parents that, like, I don't have time to drive you to school. I'm too busy at my home office. Here's the keys. Have the car drive you there. Right? There's all sorts of places this will be implemented. But still, the highest and best use. I think the number one use is still going to be drunk drivers.
The interesting thing that I looking at this though, is the fact that they've driven unplanned drives. So if you have a planned drive, you basically have a route that's been decided on, and Google probably knew exactly when lights were going to be timed. They knew exactly what was going to be going on, and they planned for all things that could happen. Unplanned drive, taking that self-driving car, that autonomous vehicle on the road to wherever. I mean, that there were unplanned drives that nobody knew about, that no city regulators, no road regulators, that they just took this vehicle out on the road, said, drive me here. And the car did that. And, that it was successful. I mean, that's the kind of test of how good the technology is, because that's the concern. Can an autonomous vehicle drive well in an unpredictable environment? It's one thing if all the cars are autonomous, but when some are and some aren't, that's crazy.
Who's going to love this is lawyers and insurance companies.
Yeah.
Because who's at fault? If you run into somebody and your car was driving, if your car didn't have the proper speed limit programmed in for that stretch of road, or maybe the city changed it and didn't update the company that pre-programs the cars, whose fault is it that you were going 50 miles an hour and what was a frontage road is now a school zone? It's Google's fault,
and we're going to sue Google with the bottomless pockets and make things right for my client who has been harmed by Google's negligence.
There you go.
Google has to pay. Very good point, Justin. Very, very good. I had not thought. Here I was thinking, imagine ending fatalities of drunk drivers because you get in your car and you're sauced. You hit the button that says, I'm too effed up to drive, and the car takes care of everything for you. But you see the beauty in the American Bar Association lining up like a pack of piranha to take bites out of the ass of Google. That's fantastic.
Well, no, but the other thing is, if you know the car is being operated in this very systematic way, and we have enough of these vehicles on the road that this is the system in which vehicles are driven. If you're driving your car yourself and you get in an accident, it's pretty much got to be your fault. If you get hit by a car, you must have jumped into the crosswalk because otherwise the car would have been able to detect you. It's one of those things, too. It's what do you trust, the person versus the computer? And I think they're going to go with eventually the computer will take over because that will be the accepted. And it'll be no fault if the computer messes up because that can just happen.
We already know that people's memory is 100% fallible. That the memory of people can be altered over time. That the things that we remember about situations are very often not correct. Mm-hmm. So it's and one person remembers something different from another person, and yeah.
So, that's great too. No, I'm sorry.
But then, do we just trust the technology?
Yes, we trust the technology.
The black box from every car. That's what we're working on. Technology. Oh, Big Brother, here we come. Oh, Big Brother.
Oh, we're
Big
all
Brother,
ready.
You know who's going to really like this technology? The robots. That's right. The robots get in their cars. I'm already spooked out by electric cars that get within like 10 feet of you before you even know there's a car anywhere near you. You can't hear them. So we're going to take these spooky-ass ninja electric cars. We're going to put the robots who are interested in world domination in these cars. And now you've got a mobile fleet of lethal, silent robot ninja assassins. This is not good for anyone. Biological. No biological people will like this. No.
That's why I'm on the robot side. World robot domination. Yay!
Here's another one then. So you don't have to drive. You've got an entertainment system. You have to have an entertainment system in the car. You can be playing video games, anything else like that. And then maybe we get to the point where there's even like not necessarily a free car, but you can get a subsidized car. You can get a car thousands of dollars cheaper if you buy this specific one, which is going to play commercials at you whenever you're in it. Like, or you know, for some period of the time then it'll interrupt your video game, it'll interrupt the movie that you're watching. You save thousands of dollars on this vehicle. It's the easiest vehicle to get into, but you got to sit through commercials.
This car is sponsored by, and we are at the end of the show. This has been a lot of fun. Scott Sigler, thank you so much for joining us today. This has been just great.
Thank you very much for having me. This is a book that will make you happy. That's all I have to say. That's it.
Nocturnal, everyone. Nocturnal is Scott Sigler's new book that's coming out April 3rd.
April 3rd.
Did I get that right? April 3rd.
However, now I've decided in a career change. It's April 3rd, scottsigler.com/nocturnal. However, if you are a lawyer and you want to sue Google, I would like to be a part of that.
Whatever you got, I'm for it. I've been harmed in that way.
You say, I'm in. I'm in. All right, everybody. On next week's show, we have Chris Mooney with the Republican Brain. That's right. Republicans and brains. I guess. That's what we're going to talk about. And shout outs, everyone. My big shout outs to Scott Sigler. This book's going to be awesome. I hope everyone gets a chance to take a look at it. And that's the end of the show.
Thank you, everyone, for listening. We hope you enjoyed this show. Twis is also available as a podcast. Just Google us in the iTunes directory, This Week in Science. Or if you have an Android device, you can look for Twis for Droid. That's Twis, the number 4 Droid, in the app marketplace. We're also, of course, in the iPhones marketplace as Twis, TWIS. For more info, wait, it's your part.
Hello, where'd you go? Oh, that's my part.
Yeah, that's.
I thought you were going to say something else.
I was starting to read your part too.
I was very confused for a moment there. For more information on anything else that you've heard here today, show notes are going to be available at our website, which is twis.org. And we also want to hear from you, so email us, Kirsten@thisweekinscience.com or Justin@thisweekinscience.com.
Be sure to put Twis, TWIS, somewhere in the subject line, or your email would be spam filtered into oblivion. You can also contact us on the Twitter at Dr. Kiki or at JacksonFly. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, please let us know.
And we're going to be back here next week, same time, same place. And we really hope that you'll join us once again for more great science news.
And if you've learned anything from today's show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got One disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy, jeopardy. And this week in science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.